The nucleus controls the activities of the cell. Each cell normally only has one nucleus. Muscle cells, osteoclast cells, and some liver cells can be multinucleated. The larger the cytoplasmic mass to be regulated, the more bosses you need. Red blood cells, RBCs, are anucleate. That is, they don't have a nucleus at all. As a result, red blood cells cannot reproduce. There are three basic parts to the nucleus. There is a nuclear envelope. This is an outer double membrane that's continuous with the endoplasmic reticulum. The nucleolus, and usually there's more than one, and the plural of nucleolus is nucleoli, is located inside the nucleus, and this is the site of ribosome synthesis. And then the chromatin is the DNA and the associated proteins known as histones. The DNA and the histones make up the chromosomes. So as we look at the nucleus, you can see this double membrane, and you can see how the endoplasmic reticulum comes off of it. So this is part of the endoplasmic reticulum. Here is a nucleolus. Usually there's more than one in the nucleus. And this is the chromatin. Now normally the chromatin is what we say condensed, so you don't see the linear chromosomes. It looks kind of diffuse here. Notice that there are pores in the nuclear membrane. Things do not freely pass in and out of the nucleus. These pores are like guards. Only certain things get in and only certain things get out. Everything has to be tagged in such a way that it can go through one of these nuclear pores. The DNA is a double helix. The histones are these circular proteins that we can kind of wrap it around. Then it will coil even more, and then it would twist and wind up until we get these chromosomes. If we didn't have this structure of wrapping and coiling, we would have long threads that would tangle up very easily inside the cell nucleus. Cell growth and reproduction is part of what the nucleus controls. The cell cycle has two basic phases, and the cell cycle is basically the life cycle of the cell. Interphase is that phase where the cell grows and undergoes normal functions. It's basically the cell being a cell. The cell division portion is known as mitosis. The cell begins division by first making two nuclei so that we have the same genetic information to pass on to each of the two new cells. And then we separate into two cells. This is the division of the cytoplasm and the organelles, and this is known as cytokinesis. So here we see the cell cycle. And notice that most of the time is spent in interphase. There are three stages to interphase. The G1 phase is the cell growth phase. This is truly the cell being a cell. There is this checkpoint that occurs, and if we pass this checkpoint, the cell is going to move on into the S phase. This is a phase of growth, but this is when we replicate the DNA. This is when we make new copies of the chromosomes. So once we get to this phase, we know the cell is going to divide. Then the third phase is a second growth phase. Here the cytoplasm is increasing and the organelles divide so that we have enough cell parts when we undergo cytokinesis. There is something called a G sub O phase. Basically, this is when a cell gets stuck in this G1 phase. If it never goes past this restriction checkpoint, it just stays a cell doing what it's supposed to, then it stops dividing. Some cells enter this G sub 0 phase when they mature. So here is interphase. We're just a cell hanging out being a cell. We have these centrosomes with its centrioles. We have the nucleus. We see the chromosome is not condensed. It's just kind of stringy there, plasma membrane. Nothing exciting going on here. DNA replication occurs during the S phase. This is when DNA makes a copy of itself. You'll remember that DNA is a double helix, so the first thing we have to do is uncoil it, and an enzyme called helicase does this. As it uncoils it, it also breaks the hydrogen bonds and unzips it. We then have another enzyme called DNA polymerase that position and link the nucleotides. Now we can make an exact copy of DNA against half of the old molecule because of something called complementary base pairing. Adenine nucleotides will always pair with thymine nucleotides, and cytosine nucleotides will always pair with guanine nucleotides. Each nucleus that we produce here will have a DNA molecule that will be half of the old original molecule and half of the new molecule. So here we have a DNA strand, and it's being unzipped by the helicase, and we're exposing these nucleotides. Now polymerases find the free nucleotides and bring them to this strand and attach them using that complementary base pairing. Adenine always with thymine, cytosine always with guanine. We end up with two strands of DNA. Now this is part of the old strand, and this is part of the old strand, and we're making a new strand and a new strand. So we're going to end up with two DNA strands identical to the original and identical to each other. Cell division is essential for growth and repair of tissues. How fast cell division occurs varies by cell type. Epithelial cells divide the most. They're pretty much constantly dividing. They can divide pretty much once a day. Nervous, cardiac muscle, and skeletal muscle cells lose their ability to divide. As they mature, they go into that G sub-zero phase and never pass that restriction checkpoint. Therefore, once these cells are damaged, we can't replace with regular cells. We have to replace with scar tissue. The M phase, or mitosis, is the phase of nuclear division. We've just made those chromosomes, and we want to make sure that one of each pair goes into the new cell. The nuclear division is divided into four phases: prophase, metaphase, anaphase, and telophase. At the end of telophase, or sometime during telophase, we have cytokinesis occur. This is when the cytoplasm and the organelles divide. We end up with two new identical cells. These are clones of the original cell. In prophase, we see the chromatin condense into chromosomes. We begin to see the linear chromosomes. The chromosomes have been duplicated, and they're held together by a centromere. So these sister chromosomes are now called chromatids. The nuclear envelope disappears, and the chromosomes are sort of dumped out into the cytoplasm. The centrosomes start moving to the opposite poles of the cell, and they begin to produce spindle fibers and aster fibers. So here's an early prophase. You can sort of see the linear chromosomes developing. And here is a late prophase where you can see the chromosomes. Notice that the nuclear membrane is gone. We have the centromeres at either end of the cell. The aster fibers help anchor them to the plasma membrane, and the spindle fibers are going to attach to the centromeres on the chromatids. In metaphase, the chromosomes align at the center. They line up on the equator. The spindle fibers are firmly attached to each centromere. In anaphase, the centromeres divide. Now we have individual chromosomes, and those spindle fibers, which are microtubules, begin to shorten and pull the chromosomes to the opposite poles. In telophase, we sort of have the reverse of prophase. The spindle fibers begin to disappear. The nucleus reforms. We begin to get a nuclear membrane around it. And cytokinesis begins. You see the cell membrane starting to come together, so we'll pinch off two new cells. This area where the cytoplasm begins to pinch together is known as a cleavage furrow. Eventually, it will join, and we will have two new daughter cells exactly like each other and exactly like the original cell. Cell division is controlled by a series of go signals. In G1, we have a checkpoint called the restriction point. This is the most important. If you don't pass this, then the cell is in G sub-zero. This cell will no longer divide. We have another signal late in G2. This is the M phase promoting factor. This is required to enter into mitosis. If the cell does not pass this signal point, it won't go into mitosis. We do have some other control signals. There are certain genes, for example, that control cell division. These repressor genes will inhibit cell division. There is a gene called p53. This gene is supposed to repress cell division, particularly when cells are crowded together. In some cases of cancer, the p53 gene is defective. This is what allows cancer cells to continue to divide even though they're crowded, and they should be getting signals to not divide. So here you see the G1 restriction point, and then here is the G2, the M checkpoint. Cells have to get here to enter into mitosis. DNA is the blueprint for making proteins. In protein synthesis, we have to convert the DNA code, that nucleotide sequence, to a protein, which is an amino acid sequence. Genes are simply segments of DNA that code for one protein. In other words, a gene is the instructions to make a polypeptide. The genetic code is 3 nucleotides. That makes a genetic word. And these 3 nucleotides, when they're found on DNA, are known as a triplet. So just like a specific arrangement of words makes a sentence, a specific arrangement of triplets makes a code that codes for one particular protein. The nucleotides make the letters of the word: adenine, guanine, thymine, and cytosine. Now, DNA is the basic code. It's the master blueprint. You never take the master blueprint to the construction site. Plus, the DNA can't leave the nucleus. So we're going to need to copy the code from the DNA. And that's going to be the function of RNA. RNA will copy the code and carry it out into the nucleus and into the cytoplasm to the ribosomes. Also, RNA will allow us only to copy one gene, not the entire genome. We're going to use complementary base pairing again. Now, RNA differs from DNA in that uracil is substituted for thymine. So wherever thymine would bind, we'll have uracil. And RNA has the sugar ribose instead of deoxyribose. RNA also tends to stay a single strand. So we're going to convert those triplets to a 3 nucleotide sequence on RNA. Now we're going to call them codons. One codon codes for one amino acid. Because we have more words than we need, we have what's called a degenerate genetic code. These are the codons from messenger RNA. You can tell because uracil is involved. But notice how we have multiple codes for the same amino acid. There's only one code for start. And we do have 3 codes that say stop here. This means we'll stop making the polypeptide chain. There are 3 types of RNA that are involved in protein synthesis. The messenger RNA, or mRNA, carries the instructions for building the protein from the gene, from the DNA, to the ribosomes in the cytoplasm. This is what we're going to make in the nucleus. The ribosomal RNA is a structural component of the ribosome. That, along with the tRNA, helps translate the message from the mRNA. The ribosomal RNA will be involved in connecting the ribosome to the correct end of the messenger RNA and in reading the messenger RNA. The transfer RNA, or the tRNA, helps bind to the amino acid, and then it will have what's called an anticodon. It will have the complementary base pairing to the codon. Based on its anticodon, it will bring a specific amino acid to the ribosome and hold it in place until that amino acid can be added to the polypeptide chain. There are basically 2 steps to protein synthesis. The first is transcription. This is when we make our copy of RNA. We make our messenger RNA. And this occurs in the nucleus because that's where the DNA is. Then we have the translation step. This is the actual protein synthesis, and it occurs in the cytoplasm. When you translate something, you change it from one language to another. Well, that's what we're doing here. We're translating from nucleic acid sentences to amino acid sentences. Transcription occurs when the DNA is separated. Again, we have a helicase that unzips it and opens it up. One side of the DNA is a template for synthesizing the complementary RNA strand. Messenger RNA is synthesized from the DNA strand, again based on complementary base pairing. Here we have an RNA polymerase that finds the appropriate nucleotide, plugs it in, and then goes back through and checks for errors. We sort of have a proofreading polymerase here. Once the messenger RNA strand is complete, the messenger RNA carries the copied gene in the nucleotide language to the cytoplasm. So the messenger RNA has to leave the nucleus. We have our DNA. We have an RNA polymerase that has to find the start point. It sits down. It goes through and it goes down the DNA, unzipping it and making complementary base pair matches to the DNA strand that it's copying. So here we have the unwinding DNA, and we have the adenine, thymine, guanine, and uracil being added or paired to the appropriate bases here. This is how we're going to get our messenger RNA. Uracil will replace thymine in this process. The DNA contains some areas that don't code for protein, these non-coding sequences. When we make the messenger RNA transcript, we copy everything on the DNA, including some of these non-coding sequences. So our first messenger RNA transcript is sort of a rough draft. We have to edit out the non-coding segments, and then the edited messenger RNA gets tagged so it can leave the nucleus. We have to let those nuclear pores know it's okay to let this molecule out. It's the edited messenger RNA that enters the cytoplasm and begins the translation step. So we have our transcription. We get our rough draft messenger RNA. We get it edited. It's now all cleaned up, it's tagged, it can go through a nuclear pore, and it comes out here so it can attach to a ribosome and start the translation step. In the translation step, the messenger RNA binds to the ribosome. Now the ribosome exists as two subunits, ribosomal RNA and a protein subunit that carries the enzymes necessary to stick amino acids together. Transfer RNA will bring the correct amino acid to the ribosome. Transfer RNA is constructed in such a way that one end of the transfer RNA will bind to an amino acid, and the other end has an anticodon. This anticodon is the complement base to the codon on the messenger RNA. So if the codon is AUA, the anticodon would be UAU. In this way, the nucleotides are translated to amino acids. So we have our gene that's copied off the DNA, and we get our messenger RNA. Here's the triplets. We get our messenger RNA and get codons. And then here's our transfer RNA and the anticodons. So AUG, that codon, would have UAC as its anticodon, and that particular transfer RNA will only attach to methionine. This particular anticodon will only bring proline, and so forth. So the transfer RNAs, based on their anticodon, will only pick up particular amino acids. And in this way, we'll be able to translate the nucleotide sentence into an amino acid sentence. As the ribosome attaches to the messenger RNA, we've got three sites here. The first site is the exit site. When something gets down here, it's kicked out of the ribosome. This site is where we have the transfer RNA come in. In this third site, we have the second amino acid come in. So we now have two amino acids side by side. The enzymes in the ribosome will peptide bond these together. This transfer RNA no longer has its amino acid attached. Its amino acid has been attached to this chain, to this amino acid, which is part of this chain now. So this one moves down here and gets kicked out. We have our chain attached here. We're ready to bring in another transfer RNA that matches this codon, and we will attach the amino acids, liberating this one, kicking it out, moving down. So we read a codon at a time as we go down this messenger RNA. So with transcription occurring, we get our pre-mRNA, which is edited. It gets to leave once it's been edited. It attaches to the ribosome. The transfer RNA, using precise base pairing, codon to anticodon, brings the appropriate amino acid in place. And we have this chain of amino acids that are made exactly according to the code. And we get a particular polypeptide chain. This will then go through the secondary, tertiary, and possibly quaternary changes to become a very specifically shaped protein. Now, reading this genetic code precisely is very important. Changing one amino acid in this chain can totally change the structure of the polypeptide that's made, or the protein that's made. There is only one amino acid difference between normal hemoglobin and the hemoglobin S that's found in sickle cell anemia. All body cells will contain the same DNA, but cells are not all identical. Various chemical signals in the embryo will tell cells that they need to change into specific developmental pathways. Some genes are turned off, others are turned on, so that while everybody has the same genetic code, they're not using all of the code. The development of these specific and distinctive features of cells is called cell differentiation. We all started out as one fertilized egg, one cell with all the same stuff. And for several weeks, all of the cells were identical. But very shortly, we had cells that were destined to become muscle cells, other cells destined to become skin cells. That's the process of cell differentiation. They all still have all the same DNA, but they're not listening to all of their DNA. Some of that DNA has been put on hold. Apoptosis or apoptosis is programmed cell death. This can occur when cells are no longer useful, when they're aged. If cells become too crowded, some of them may die. In the developing embryo, we have some cells that we need only briefly, and then they disappear by apoptosis. Hyperplasia occurs when we have rapid cell growth. When someone has anemia, they don't have enough red blood cells, the bone marrow will undergo hyperplasia, rapid cell growth, to make more red blood cells. An atrophy is a decrease in the size of an organ or a body tissue. Muscles that lose their nerve supply will atrophy and waste away. In polio, muscles lose their nerve supply. When this happens, they're no longer told to contract. This will cause muscle cells to atrophy and waste away. If you've ever had a cast on your arm, when the cast first came off, you may have noticed that the casted arm was a little bit smaller than the arm you'd been using all along. Your muscles hadn't atrophied completely, but they were beginning to lose some muscle mass because you hadn't been using them very much. No one really knows what causes aging. That's still a mystery, but there are several theories on why we age. One is the wear and tear theory. This one simply says that there's a whole lifetime of chemical insults and free radicals that cumulatively beat up on the cells and they just get old, they wear out. There's the mitochondrial theory of aging. Because the mitochondria are the powerhouse of the cell and we need that energy to drive cellular reactions, the idea here is that free radicals in the mitochondria diminish their ability to produce energy, and so if we can't produce energy, we just don't have the cells that we need. Another theory is that there are immune system disorders. Remember, your cells all have that glycocalyx and those receptors and all of those biological markers. Your immune system is supposed to identify those as yours and not bother them. However, in autoimmunity, the immune system decides that your normal biological markers are the enemy, and they will begin to damage cells. So this could lead to a progressive weakening of the immune response as well as damage to cells. However, the most popular and widely accepted theory is the genetic theory, the idea of the telomere clock. Whenever you replicate DNA, you have to have those enzymes sit down to start the replication process. Wherever those promoters sit, those 2 or 3 nucleotides don't get copied, which means every time you copy your DNA, the DNA molecule gets a little shorter. The cell aging theory using the genetic theory claims that the cessation of mitosis and cell aging are programmed into our genes. These strings of nucleotides that are at the ends of the DNA molecule are called telomeres, and so they don't code for anything. If we lose little bits of our telomere, it doesn't really affect how the DNA works. But as cells divide, a little more of the telomere comes off and the chromosome gets shorter and shorter. Once the chromosome gets to a certain length, once it gets to a certain shortness, then there is a stop signal and the cell will no longer divide. If we can't replace damaged cells, then we're going to show aging.